Welcome to the all viewers and well-wishers of the word. The purpose of my lecture is learn the knowledge from the word and return the knowledge to the word. Motto of my life is learn, earn and return. I have learned a lot, earned a lot, and now to return a lot to the word from where I have got all this knowledge. The topic of today is antibiotics, or in the broader term, one should say like that, it is antimicrobials. But I am going to talk today is the basic of antibiotics or antimicrobial. So the antibiotic are the chemotherapeutic agents. Term antibiotic is used to describe antimicrobial agents, usually antibacterial, used to treat infection. This term is wrongly used. The term antibiotic strictly means substances that are of biological origin. The chemotherapeutic agent referred to a synthetic chemical work as antimicrobial. With advent of the synthetic methods, this definition has been modified. Newer antibiotics are chemically modified biological products or chemically synthesized biological products. The generic term for these two are antimicrobic or antimicrobial agents, abbreviated as AMA, but usually antibacterial. So here one can see the antibiotic which are being used in the form of capsules, in the form of tablets, in the form of injections, the powder and syrups. So all these are the form of antibiotics. If bacteria cross our immune system, they get inside our bodies, they cause disease. But the chemical substances that inhibit growth or cause death of the microorganism in our body, these are antibiotics or antimicrobials. Antibiotics are the chemical produced by a microorganism that kills or inhibit growth of another microorganism. So they are of biological origin. These are selective poison. They kill bacteria but not harm the body cells. Different antibiotics kill different bacteria in different ways. Antibiotics are specific to the certain bacteria and capable of killing the specific bacteria. They have in vitro and in vivo selectivity for the bacteria and sparing the host cells. They work more efficiently in conjunction with an active immune system, and without immune system they are not capable of doing that harm to the bacteria as with in conjunction with immune system. So the antibiotic or antimicrobials, they are naturally occurring, produced by the fungus or the bacterium as the secondary metabolite. They do not kill those bacteria which produce them, but they are capable of killing the other bacteria which can harm the body. Semi-synthetic, synthetic derivatives of the naturally occurring antibiotic. We get the formula of these naturally occurring antibiotic and make in our factories. So these are called semi-synthetic. They are made by the chemical manipulation of the naturally occurring compounds in our pharmaceutical factories. Now, synthetic or chemical, these are not naturally occurring, these have not been found in the nature, but they have been made by the man. So these are the man-made antimicrobial compounds. These are not occurring as the fungus or the bacteria are going to produce. So examples are sulfonamides, quinolones, nitrofurans, and imidazole are the strictly chemotherapeutic agents, not naturally occurring. So here there are certain bacteria and the fungi which are capable of producing the different antibiotic. Here one can see the Bacillus subtilis is able to produce acetylcysteine. Bacillus polymixa is going to produce polymyxin. Similarly, the fungus Streptomyces that is going to produce amphotericin B, and the other streptomycin when usually these are capable of producing chloramphenicol, and some are going to produce chlortetracycline and tetracycline that is also being produced by the fungus. Similarly, streptomycin erythroase that is capable of producing erythromycin. Neomycin is being produced by streptomycin fredii, and the streptomycin is being produced by the Streptomyces gracius, and Micromonaspora purpura is able to produce the gentamicin. Similarly, fungi, Cephalosporium species, they produce phyllothin, and Penicillium glycofulvin is capable of producing glycofulvin, antibiotic and antifungal, and similarly the last is the Penicillium notatum is producing the penicillin, which has revolutionized the antibiotic era. Now, history or the discovery of the antimicrobial drugs, different preparations were applied to the wounds to destroy infection. Indians used the bark from the cinchona tree to treat malaria. Similarly, in China, poultices used contain antibacterial and antifungal substances. In 19th century discovery, one microorganism is capable of destroying another species, so the modern chemotherapy dates to the German chemist Paul Ehrlich in 1854 to 1915, who used systemically effective agents to treat trypanosomiasis and the syphilis. The drugs were given systemically to destroy all these pathogens. They discovered pro-rosanilin, antitrypanosomal, and R-sulfonamine effective against syphilis at that time. So that was the Ehrlich's magic bullets he has made and fired against the bacteria, and here that is magic bullet which is capable of killing the different kind of the bacteria. All the credit goes to this gentleman who have just contributed a lot towards the antibiotic word. Now, continuing the history, Paul Ehrlich developed idea of the selective toxicity magic bullet concept, that is sparing the host cell and killing the bacteria. In 1932, Gerhard Domack discovered red eye prontosil, first sulfur drug, that is a chemically produced drug. Penicillin G was discovered in 1929 by Great Fleming. In 1944, Salman Waxman and Albert Schatz, American microbiologist, isolated streptomycin from Streptomyces gracius fungus and other antibiotics such as chloramphenicol, tetracycline, and erythromycin in soil samples with the fungus, and they produced all these antibiotics, got it extracted from the fungus. This is the Fleming and the penicillin. If the fungus he has grown that contained the penicillin that was capable of killing the other bacteria on the petri dish. So it was thought, and he discovered that this fungus contains a substance which is called penicillin that is capable of killing the many gram-positive bacteria, and so this contribution is a great contribution. Now, discovery of the antibiotics, if you just look at this, in 1940, beta-lactams and the sulfur drugs were discovered. In 1950, macrolides and aminoglycosides and chloramphenicol and tetracycline were discovered. In 1960, the glycopeptide and the quinolones and streptogramine were discovered. But after that, there was a long gap from 1960 to 2000 when the other molecules were discovered, that is oxazolidinone and that is lipopeptides and the other mutilins. These were discovered in 2000 and 2010, but there was a lot innovation gap, and this gap have led to the bacterial resistance, and they were not responding to these previously discovered antibiotics. Now, no expectation, no disappointment. So expecting is my favorite crime, as most of the human beings do, and disappointment is always my punishment, which one get. But I do say, if no expectation, no disappointment, you don't feel hurt because you do not expect. Zero expectation. If someone do good for you, even 1%, 5%, 10%, that is a gain and that gives you the happiness. But disappointment. Now, chemotherapy is selective toxicity. Antibiotics are more toxic to the invading microorganism than to the human host. There are certain reasons for that, which I am going to tell you. Why do they spare human cells and kill the bacterial cells? Reason is, bacteria have cell wall composed of the peptidoglycan and ribosome 70S. We make the antibiotics which are capable of destroying this peptidoglycan and ribosome, which is of 70S nature, because human ribosomes are 80S nature, and these are being spared by the antibiotics, and the peptidoglycan is the unique in the bacteria, so it is not found anywhere in any other tissue. So these are being killed by these antibiotics which will fire against them. A human cell do not have peptidoglycan. Similarly, ribosomes are of 80S nature. This is called selective toxicity that distinguishes antibiotic from disinfectant. Just to have an example, if the army have been asked to kill the people, those who are wearing the red caps, and sparing those who are wearing the green caps, so that is called selective killing and selective toxicity. When selectivity is high, antibiotics are not toxic, but still in very high dose, they can have some side effect. As you want to eat the good thing, after some time, this good thing is going to be the bad thing. Except sweetening is also sometimes not good. So here one can have the idea that the bacterial cell, they have got a cell wall, and that is covering the cell membrane, and this cell wall is made up of peptidoglycan. And if you just want to have the detail, you can have my bacterial anatomy lecture in which I have told a bit. So peptidoglycan is basically structure is that is the glycan spore, which is just like a ladder, and these are connected by the peptido bonds, and these antibiotics, they just attack here and break that junction. So these glycan strands become attached, and these are just like the bamboos. These are not capable of giving any function. Just like the ladder, if the connecting sticks between the two main ladder bamboos are broken, the result will be of no use. The ladder will not be of any use. Same is the case here with the antibiotic attack, the peptidoglycan. Similarly, the bacteria are capable of killing 70S ribosome, that is prokaryotic ribosome, and they are not capable of killing eukaryotic ribosome. These are of 80S nature, so these are spared and these are being killed. So this is all called selective toxicity. Now, disinfectants, antiseptic and antimicrobial. Disinfectant and antiseptics differ from the systemic antimicrobial. They possess little selective toxicity and they are capable of killing any bacteria anywhere found. So these are toxic to both microbial pathogens but as well as they do damage the host cell. It can be used only to inactivate microorganisms in inanimate environment like fomites, furniture and other things, but to some extent on the skin surface, but it cannot be taken systemically because they are not capable of killing the bacteria but also killing the host cell. The antimicrobial action of the disinfectant depend upon the concentration, time and temperature. So mostly they are surface acting but they cannot be given systemically. Now, guidance of the antimicrobial therapy is minimum inhibitory concentration. As the name is, the lower concentration of any antibiotic that is capable of inhibiting the visible growth but not capable of killing the bacteria altogether. That is minimum bactericidal concentration, MBC, lowest concentration of the antibiotic that kills nearly all or 99.9% of the inoculum of the bacterium. So, blood concentration of the antibiotic must be above the MIC and MBC of the bacteria, then one is able to get the killing effect of the antibiotic. Then therapeutic index is the toxic dose or the effective dose of the antibiotic, and categories of the antibiotics are bactericidal and bacteriostatic. I will tell you in next slide the detail of that. So here one can see we are increasing the antibacterial concentration and they are not capable of growing any bacteria on the culture plate. But when the concentration of this antimicrobial or antibiotic is less enough, that is just allow the bacteria to grow, so there is growth of the microorganism at this lower concentration of the antibiotic. So this is called MIC, minimum inhibitory concentration. But as we just go more forward, increasing the dose more, we are just having the no growth on the culture plate. It means this quantity of the antibiotic is sufficient enough to kill all the bacteria and does not allow any bacteria to grow on the plate. So this is called minimum bactericidal concentration or MBC. Same is the case here. As one can see, the bacteria here, the lesser concentration bacteria can grow, but here the bacteria cannot grow here. So this is the minimum inhibitory concentration where the bacteria stop multiplying and growing, but they are not going to be killed. And here one can see the MBC, that is 32 microgram per ml, the concentration at which the bacteria cannot grow, and these are all killed altogether. So here is the difference between MIC, the 4 microgram, and the MBC 32 microgram. Here you have also seen, so one can make it understand more easily. Now, what is bacteriostatic? Static means to make anything static or stationary. So, by definition, at usual dosage, they prevent active multiplication of the bacteria, but they do not kill. Examples are chloramphenicol, tetracycline, and erythromycin. They inhibit growth of the bacterium but not kill them. The bacteria can grow again when the drug is withdrawn or the effect of the antibiotic is no more. The bacteria grow again. Bacteriostatic agents are beneficial in the sense that they use the host defense mechanism with them to kill the bacteria, so such as phagocytosis, to destroy or to kill the bacteria. So the duration of therapy is longer. Some bacteriostatic agents in higher concentration act as the bactericidal effect, like erythromycin and tetracycline. Bactericidal, cidal mean capable of killing. So that is bacteriostatic in higher doses can work as bactericidal, but we have to consider the toxic effect of that dose and the side effect of that dose. Instead of getting the bactericidal effect, we also have some side effect. So the next is bactericidal. At usual dosage, they are capable of killing the bacteria. Examples are penicillin, cephalosporin, glycopeptides, and aminoglycosides. Bactericidal drugs are more effective, particularly useful in certain infection, life-threatening infection, whose when polymorphonuclear leukocytes or TLC count is below 500 per liter and the suspicion of endocarditis where phagocytosis is limited by the fibrous network of the vegetation and bacteriostatic drugs do not affect a cure. They are not capable of having that killing effect. So we have to use the bactericidal drugs. These are also used for the meningeal affection, that is meningitis, that is going to be a life-threatening problem. Here one can see the bacteria, if no antibiotic has been applied on that, they just do merrymaking and grow very rapidly and multiply. Now, we have used the bacteriostatic drugs. Here one can see we have bound them, we have roped them, we have just locked them that they are not going to be multiply as like this, and but they bacteriostatic antibiotic prevent the multiplication, but they are not capable of killing. But here, if we apply or we give bactericidal antibiotic, they are not only stop the multiplication but also kill the bacteria, so they are not capable of multiplying again. Similarly, in the bacteriostatic and bactericidal action mechanism, no antibiotic, bacteria multiply. Bacteriostatic antibiotic prevent multiplication, and bactericidal, they make the bacteria die. It is just the replication of this diagram. Now, broad spectrum antibiotic, effective against a wide range of microbes, both gram positive and gram negative. Examples are some lactam antibiotic, tetracycline, aminoglycoside, sulfonamides, and chloramphenicol. These are broad spectrum. Tetracyclines are active against many gram-negative rods, atypical bacteria like chlamydia, mycoplasma, and rickettsia. And narrow spectrum, they are effective only against a narrow range of microbes, either gram-positive or gram-negative. Those with activity against one or few types of the bacteria, like vancomycin antibiotic works against staphylococcus and enterococci only. It is better to use narrow spectrum antibiotic if infective agent is known to you, as this will not harm normal bacterial flora, and we are not going to use the broad spectrum antibiotic. Here one can understand narrow spectrum antibiotic act against a limited group of bacteria, that is, they work only one kind of bacteria, that is the green color bacteria. But broad spectrum antibiotic act against a larger group of bacteria. They can work against gram negative, gram positive, cocci, bacilli, different nature of the bacteria. And to understand in a common word, the broad spectrum is like a cannon, it fires, and it is not sometimes capable of killing the enemy. But this rocket launcher is better, though it is narrow spectrum, but it is capable of killing the life-threatening single type of the bacteria more efficiently than this cannon. Now, in vivo affecting factors, certain factors which affect when the antibiotics are given to the human being. Activity of the antimicrobial agent in vivo is different than in vitro, that is, in the petri dish or in the test tube. Analysis of the activity of the antimicrobial agent in vivo is much more complex than in vitro, that is, in test tube. The activity too involves drug and pathogen, but also the third factor, which is the host, in which all this is happening, that is the battlefield. Drug pathogen and host pathogen relationship are different, and I will discuss all these one by one. Host drug relationships are absorption of the drug, excretion, distribution, metabolism of the drug in the liver or the kidney, and the toxicity or the side effect of the drug when it is being used by the human body. Abscess, circulation is blocked off and antibiotic is not able to reach there. Similarly, foreign bodies, obstruction of the urinary bladder, biliary or the respiratory tract, etc. These are also having the no reach of the antibiotic properly. Perfusion is not as good as it should be. Similarly, immunity is also one of the factors which affect in vivo. Now, drug pathogen relationship, number one factor is environment. In host, different environmental factors affect the microbe located in the different tissue and the part of the body, because the different body tissue behave differently and their metabolism is different. In contrast to the test tube or the petri dish where their factors are constant, the environment is same. Response of the microbes is not uniform as in the test tube. Number two factor is absorption. The absorption of the drug from the intestinal tract, if taken orally or by mouth, or from tissue if given injection, is different. So, oral and injectable have different absorption. There may be continuous excretion or inactivation of the drug. So, drug level in the different body parts fluctuate and microbes are exposed to the varying concentration of the antimicrobial agent, and absorption orally is slower and by injection is faster. So the absorption is different. Third is distribution or concentration of drug in different body tissues. In body, antimicrobial agent is not equally distributed in tissues and fluids. Some tissue have more love and liking for the antibiotic, particularly some antibiotic, and some do not allow the antibiotic to reach to their fluid and tissues. In body, microbes are not exposed to a constant concentration of drug as in the test tube. Many drugs do not reach central nervous system or prostate effectively. They are not able to cross the blood-brain barrier, so these will not be effective for encephalitis and other CNS infection like abscess and other. So the drugs given systemically are inadequate for effective treatment here. Some drugs have more concentration in urine than in blood or other tissue, so these drugs are being used to treat the urinary tract infection more efficiently than the drugs which are given systemically. The continued distribution and concentration of drug, necrotic tissue or the pus absorb drug and prevent its contact with the bacteria. I have told you that does not allow the antibiotic to reach to the proper targets. The distribution of the drugs varies greatly with the different tissues. Topical or the local drugs for wounds or mucous membrane like conjunctiva, which are poorly absorbed, provide highly effective local concentration without toxic side effect. These are effective topically more than if they are being used systemically. But some drugs applied topically on surface wounds are well absorbed and cause toxic pain. So all the topical drugs are not being absorbed. Some are notorious to be absorbed and causing the toxic effect when they reach to the blood. So here one can see the pharmacodynamics or pharmacokinetics of the drug, and we just remember it by a simple way: A, D, M, E. A for absorption, D for distribution, M for metabolism, and E for excretion. And activity here, one can see the liberation, that is L. Drug reaches to the body, being absorbed via the gut or via the injection. Then that is distribution occurring in the different body tissue, and then metabolism, whether in the liver or the other body organs or the kidney, and ultimately there is excretion of the drug via urine or via feces. So this is all pharmacodynamics. Now, number 4 is location of the organism. In body, microbes are located within the tissue cells. Drugs enter tissue cells at the different rate. Different tissue have the different liking of the different antibiotics, that is called tropism. Some, like tetracyclines, reach about the same concentration inside the monocyte as in the extracellular fluid. With other drugs like gentamicin, drug probably does not enter host cell at all. So this is in contrast to the test tube, where microbes come into direct contact with the drug, whatever it is, whether being liked by the bacteria or not. Now, the 5th factor is interfering substances. The biochemical environment for the microbes in the host body is very complex and interfere with the drug action. The drug may be bound by the blood and the tissue proteins or the phospholipase, and drug may react with the nucleic acid in pus and may be absorbed onto exudates, cells, and necrotic debris. In necrotic tissue, pH is highly acidic and not suitable for the drug action, like aminoglycoside, and these drugs are being destroyed or inactivated by the pH of the necrotic tissue. Number 6 factor is variability of the concentration. It is essential to maintain an effective concentration of the drug where infecting microbes proliferate. If the drug level will be less than the MIC or MBC of the bacteria will just be very happy and not being killed. But if the serum level of the antibiotic is more than the MIC or MBC, that will having the killing effect on the bacteria.